Hello.
Hello, everyone. Good afternoon. This is the English oral presentation session. But just to let you know that our first presenter, Dr. Amanda Windsor, will not be coming. She had a last minute emergency, so she will not show up. But we have our next presenter at 4 o'clock. So we'll give it a few minutes so she can come in and present her work for us. All right. Thank you, everyone. Hello, everyone. We're back. We have our next presenter with us. Our next presenter is Afia Sultana. She's going to present the work entitled Embryonic Development of Slipper Lobsters, Ibacus ciliatus and Ibacus novebrantatus. Afia, did you put your slides in, or do you want me to help you?
Hello, good morning. Okay, I try first to share my.
We can't hear you, I don't think.
Uh, can you hear me now? Uh, Sorry, just a moment. Can you hear me?
Yeah, no, we can't hear you. There's no sound.
Just a moment.
Maybe it's us.
Now, now can you hear me? Just a moment.
We're checking if it's on our end.
Is it okay now?
All right, so the issue is us. You're okay, it's us. Just give us a second. We're gonna solve this here.
Okay, sure.
Can we try again? I think we solved it.
Hello, can you hear me now?
No, we didn't. Sorry. Wait.
Sorry. Okay. Yeah.
Oh yeah, okay, yeah, finally. We solved it. Yeah.
Okay, good
I'm
morning.
so sorry. I'm so sorry, Afia.
It's okay. Now I am audible.
Yeah, now we can hear you. I'm sorry.
Yeah, sure. So now I'm trying to share my screen.
Okay.
Just a moment. Sorry.
No, it's fine. Take your time.
Okay. Sorry.
No, it's okay. If you have any issues, let me know and we can help.
Now my screen is visible. My slides.
Not yet.
Oh,
Do you see them on the screen? Does it show up
yeah.
for you?
Yes. Now, not still.
No. Did you share the screen by going
Yeah,
into the share
I
screen?
did. I did.
Yeah. It should show up, but we're not seeing it.
Share screen. I did share the. Okay. Enter screen. Okay. Not yet.
No.
Now, what do you see? My screen?
No. Do you want to try to upload your slides? Maybe I'm not sure if it's a problem with our internet connection. It might be.
Just a moment. Now, maybe you can
It's
see.
showing up. It's showing up. Yeah.
Uh,
Yay. Yeah.
Sorry for the inconvenience. Okay.
Yeah.
Yeah. Should
Okay.
I
So
proceed?
yeah, we can see it perfectly now. So you can put it on full screen, and you can start whenever you're ready.
Okay. So now I have put on the full screen. Is it visible?
No, it's still small for us.
Now, no.
No. Maybe if you share the full screen, or if
Yeah,
no.
I did. Why? Just a moment.
No, it's fine.
I did the full screen, but somehow it's not.
Huh.
Now it's okay.
No, we're still seeing it small. Can you try the little icon with the presenting thing? Maybe we can see it that way. It might be because it's something about the notes, maybe.
Notes. I haven't had any notes. Sorry.
Oh, I'm sorry. I'm sorry.
Slides and PDFs. Slides. Oh, sorry. How?
No, it's okay. Don't worry about it. We're gonna figure this out. Um, do you? A gente tem apresentação lá? Acho que temos. Do you want us to try and do it from our end if we have?
Yeah, it's okay. If you can share the slide for me, it's also okay. I think I have sent you the files.
Okay, yeah, we have it.
Uh,
We're trying to put it in now.
okay, sorry.
No, it's all right. I'm so sorry. I'm so sorry. It's our issue. So just let me know when you want us to go forward on the slides.
Okay.
But
Now it's
thank
okay.
you. Yeah, now we can see it. You can present whenever you're ready. Thank you.
Okay, okay, thank you so much. Okay, so sorry for all of this, first of all. So I'm Mosamata Afes Sultana. I'm a current graduate student at Hiroshima University here in Japan. And today I'll be presenting embryonic development of sleeper lobster, Ibacus ciliatus and Ibacus novum dentatus. And my co-authors are Narumi Sawada and Professor Kari Okabayashi. Okay, so let's talk about lobster. Okay, so marine lobster are very popular as highly valued seafood, at least where they are available. And skylarite lobster, a particular group of lobster that I am studying, are relatively less explored, but still they support the global demands for lobster. Okay, so here is a market analysis that shows that the lobster market will actually expand, and in next 10 years, the total value for lobster will be more than doubled. But at the same time, there are also studies that are showing that the global warming is actually affecting the wild lobster stock, just like all other animals. And like this study that is showing that American lobster stock are moving towards the Canadian water as the Canada is colder. But if the global warming continues like this, at some point of time, even the Canadian water will be too cold for them to continue to sustain their population. So what does it mean? It means that if we continue to rely fully on the oil stock of the lobster for our consumption, maybe we will just be paying more unit price for lobster. That's how the value will increase. So we have to do something with the aquaculture. We should develop the aquaculture practice for as many species as we can. So the sleeper lobster or skylarite lobster are underexplored, but still nowadays the studies are finding that they are actually gaining value as commercial alternative to other lobster group like spiny lobster or clawed lobster. Those are called the true lobster. Okay, so these two are the main character of my research, Ibacus ciliatus and Ibacus novum dentatus, and they are widely distributed from the East African waters to East Asian waters, and they are known to have a higher potential of being aquaculture target because they have relatively shorter lifespan, particularly in the larval development. They will complete it very quickly, so that's really important for the aquaculture. And my particular focus is on the embryonic development because it is a very important phase in their life cycle, and most of the foundational biological processes are completed here. And if we understand them very clearly, then using that knowledge we can optimize the hatchery practices and improve the survival rate, particularly in this young stage when they are very vulnerable. They are very sensitive, just like the babies for all other animals. Okay, so to clearly state the objective of my study was to make criteria for various embryonic stages of Imbacus ciliatus and Imbacus novum dentatus from morphological features of the embryos that will facilitate the seed production, which in turns will help us to develop the aquaculture technique for not only the sleeper lobster but also for spiny lobsters, because when it comes to embryonic development, they are pretty close, pretty similar. Okay, so our methodology was very simple. We collected the egg-bearing mother females from the market in Japan, and then we transported them. Oh, sorry. Okay. We transported them to our laboratory and cultured them in the recirculating system. And when culturing them, I tried to sample the embryo from the mother lobster and then observe them under the stereo microscope to see the morphological features and the development going on. Okay, so for my study, actually, it was very important to measure the yolk content of the embryo, and I would like to give hint here because it's really important. Like when I am taking the measurement from the photo, uh, if I use different angle and different position of the embryo, then it can actually lead us to different kind of measurement from the same embryo if we use the different position and different angles. So I had to make sure that I am use the same position for all the yolks. So I defined my position like this. From the photo, two eyes of the embryo should be visible, and only for those photo I will include those in my analysis. Like here for this first embryo, these two doesn't show the eye completely, maybe eye is at the back, and that's why the yolk is fully not visible. So I excluded this kind of photo from my analysis, and I used only this kind where the eye, we can see the eye like here and here and like this. So for all the photo should be like this. Then I will take the circumference of my embryo and then perimeter of the yolk, then I will make the proportion. So this way I did this. Okay, here is my result. Okay, for both of the species, I found that the embryonic development is pretty same, almost similar in these two species, and I defined 6 different stages for both of these species. And now I will define the stages. For the stage, the embryo is full of yolk. There is no other structure visible. For stage 2, the egg membrane separated from the embryo. That means the yolk is reducing. That's how a gap is formed between the membrane and the embryo. And when there is two eyes, compound eyes is visible, I'll call this as stage 3. And then it's in stage 4, they will have the heartbeat. Under the microscope, the heart tissue will be visible like this. There will be the vibration. So this is stage 4. Okay, after reaching to stage 4, it's actually tricky. There is no other like key morphology that we can see rather than the yolk content, like yolk consumption. So after this, we can say by the stage 4, the major development has been already completed. So after this, they will just keep consuming the yolk. So at this stage, the yolk is around 30 to 50%. So when the yolk is reduced to 30 to 10%, I will say it's stage 5. And then below 10%, I will say this is stage 6. They are ready to hatch. Okay, so this is the diameter of the embryo that ranged from around 0.9 millimeter to 1.18 millimeter in this species, and there were significant difference from stage 1 to stage 4. But when it reached stage 4, 5, and 6 doesn't have any significant difference, just similar to the development pattern for the stages. Okay. So this is the development for Ibaca ciliatus other species. The definition, the staging remains same for this species. There is some slight minor difference, like the egg diameter is slightly lower here. It ranged from 0.89 millimeter to around 1.1 millimeter. So the other thing remains same. So here I would like to conclude my presentation saying that the criteria for embryonic stages, the definition proposed here is actually a foundation for better understanding of the developmental biology for these species. And this knowledge can be used for further studies. Like for myself, I have the plan to study the developmental duration and different temperature setting using these criteria so that I can predict the hatching date very correctly. That will be very helpful for the farmers to prepare for the next step, like the larval development. Like here is an example. Here in Japan, we have the very beautiful cherry blossom. So for the cherry blossom, there is the cherry blossom prediction that is very accurate. So that's important for the tourism industry. They can plan their trip ahead. So my study is also same. They use the cumulative air temperature for any particular region to forecast the cherry blossom, full blossom. For my study, I will use also the cumulative temperature for the whole incubation period of these species to understand how long it takes for them to incubate their embryo and when they can like hatch to prepare for the next step in the hatchery. So that's my plan. I like to talk about my research, but I think there is time limitation. So thank you all for your patience hearing. Thank you.
Hi, Afia. Thank you so much. It's very interesting work. We have a question for you.
Okay, sure.
Congratulations, Afia. Have you calculated the egg loss between stages? Do you know if it is significative?
Okay. Yeah, the survival is really very important, and I didn't like very accurately made the survival rate of the embryo itself, but yeah, when I was culturing, the whole incubation period is roughly around 1.5 to 2 months. So the mother will carry the egg for this duration, and during this duration they will lose some of the egg every day. So they have like a big class like this, so they have like thousands of eggs carrying, and then every day they will drop some egg. So maybe I can say roughly from my observation, maybe 20 to 30% egg is lost, and 70 to 80% around, and it actually varies. There is individual difference, so it will vary. And we are just trying to culture this lobster. Like there is no aquaculture for this lobster, so we are the first on to trying to culture them in a lab condition. So for now the survival rate is not very great, I will say. For some
Like,
of the
yeah.
lobsters they will hatch successfully.
Yeah, you said that it has like thousands of eggs. Do you
Hmm.
know how many eggs they usually have?
Okay, for now I don't really know exactly the fertility, how much, but yeah, sorry.
No, that's okay. I'm sure there are a lot of eggs.
Yeah, they have.
Oh, and you said you guys bought them from the market. They sell them alive?
Yes, they sell them
Okay,
alive.
yeah, that's very interesting. We
They will
have
just
some.
hi.
No, I'm
Yes.
sorry. Yeah, I'm just gonna say that we have some here that are sold from other species, and they are usually not alive on the markets.
Okay.
We
Hmm.
have another question. Is the development of these species different from that of Palinurus? If so, is a new protocol needed? Do you know?
Yes, I think Phanylurus is the spiny lobster, and
Mhm.
I'm talking about the sleeper lobster. So they have some basic difference in the embryonic development pattern. There is no
Okay.
study so far for the sleeper lobster, so we are studying to see the difference actually.
Yeah. Yeah, it's a very nice study. Yeah, you're having a lot
Thank
of
you.
congratulations. Thank you very much. We don't have any more questions, but thank you so much, Afia. It was a pleasure to have you. I'm so sorry for all our connection issues,
Yeah, I'm sure.
but thank
Yes.
you for participating. We're now going to call the next presenter. So the next presenter is Sofia Calderon Lopez, and she sent us a video. We're going to play that and then call her to answer questions.
Hello, my name is Sofia. I am PhD student at the Systematics and Biogeography Paracarita Crustaceans Lab at the Buenos Aires University in Argentina. Today, I'm going to present Dipsy aceleora crustacea isopoda from the Mar del Plata Submarine Canyon, Southwest Atlantic Ocean preliminary results in co-authorship with my supervisors, Drs. Daniel Rocataglieta and Brenda Dotti. The Aceleota are a diverse group of isopods. Around 2,000 species have been described so far, many of which are found in freshwater and marine environments. In the ocean, they can be found from the shallow to the deep sea. At least 11 families are present in the deep sea and have adapted to this environment through different lineages. Doty et al. 2020 compiled the biodiversity of deep sea Aceleora off the coast of Argentina in a review of the isopod, amphipod, and Comassion fauna of the region's deep sea. Regarding the phylogeny of Aceleora, Raupach et al. 2009 proposed this phylogenetic hypothesis, which is based on molecular data. The blue lineages represent taxa from shallow waters, while the black lineages correspond to taxa from deep sea environments. The authors of the study have concluded that there were 4 events of deep sea colonization in Aceleora. One of these events led to the diversification of most deep sea families, while family Acanthaspididae resulted from a distinct colonization event. Of the 4 events, 3 were attributable to species of Janiridae migration. Now, knowing that the deep sea Aceleora have shown a high diversity in other areas, then the hypothesis of this work is that the Mar del Plata submarine canyon shows a higher biodiversity of Aceleora isopods than previously known, including new species to science. And in this regard, the general objective is to update the number of families, genera, and species of the Aceleora isopods from the study area. Regarding the methodology used, the material was obtained on board RV Puerto Deseado on the service Salud Continental 1-3 during the years 2012 and 2013. In the map at my right, it is shown the stations in the Mar del Plata submarine canyon where Aceleora isopods were collected. The bathymetric range of the samples was 250 to 3,220 meters depth. For the sampling, the material was collected with a bottom motor troll and 2 epibentic sledges of 1 mm and 13 mm mesh size. Next, the material collected with the epibentic sledges were split into buckets. After the buckets, samples were sieved using a 250 mm mesh size. Most of the samples were fixed in formalin 5%, then were transferred to ethanol 70% for manipulation. A small fraction were also fixed in 96% ethanol for molecular studies. Specimens were sorted and identified to species level under a dissecting microscope. With respect to the results, a total of 1,885 ocelots were obtained and 78 species belonging to 14 families were identified. 1 species of the genus Oxylus, Incertisedis genus of the superfamily Janiridea, is reported for the first time. Also, here you can see a bar graphic representing the number of species per family found so far. Munopsidae was the most speciose family with 27 species, followed by Paramunidae with 17. The families Aplomunidae, Ichnomesidae, Dueropsidae, Catianiridae, Nanonisidae, and the genus Oxylus are represented in our samples by only 1 species each. Naecellus argentinensis and Esenetridae incertisedis erraticaudum were collected in 9 and 8 stations respectively, whereas the remaining species were collected in 1 to 6 stations. Among the species identified, at least 20 are new to science and are distributed in the following 6 families: Acanthaspididae, Dendrotionidae, Janiridae, Catianiridae, Munopsidae, and Paramunidae. Regarding Munopsidae, the genus Notopais is reported for the first time to the study area, and 3 new species of this genus were also identified. Additionally, the genus Euricope had only 1 previous record for the Mar del Plata submarine canyon, and here we are reporting 5 species of the genus. We have identified 2 new species of Dendrotionidae and 2 of Janiridae. In the family Paramunidae were found 7 new species distributed in 5 genera. For the family Acanthaspididae, we identified 2 new species of the genera Iolanthe and Iantopsis. I will work with this family during my PhD, due to taxonomy doubts about the genera diagnosis. The family Dueropsidae was found in 2 stations, and only the species Dueropsis intermedius was identified. This species is usually present in shallower waters. Molecular studies will be necessary to confirm this extension of the bathymetric range for the species. This is the first record of the family Catianiridae for the Southwest Atlantic Ocean. 1 new species of the genus Natalianira for the Mar del Plata submarine canyon was found. We also identified 1 new species of Natalianira and 1 of Catianira of the Burbot Bank. We are in the process of sending these findings for publication. In regard to the discussion, half of the species were collected only once, totaling 221 specimens and representing 12% of the total number of specimens collected. This result suggests that most of the 36 species are scarce in the study area. Future ecological studies are needed to prove different hypotheses about this phenomenon. Brandt et al. 2007 had similar results and suggest that these results may reflect a patchy distribution of these species. While the results presented here are preliminary, the ocelot and high diversity observed in the Mar del Plata submarine canyon is consistent with the high diversity found in other deep sea areas. Doty et al. 2020 reported 8 species from the Mar del Plata submarine canyon area. The results here presented exceed by far this number. Similar results were obtained by Pereira et al. 2025 with viviparous isopods. Finally, some future considerations. This year took place the Survey Talud Continental 4 on board RB Falcor 2 conducted by the Schmidt Ocean Institute. Samples were taken with a remotely operated vehicle called Sebastian and landers with baits. We are now processing these samples to do molecular analysis in the future. In addition, images of the environment also need to be processed. During the transmission with the ROV, the different environments of the Mar del Plata submarine canyon were seen for the first time. Associations between paracarida and ecosystem assemblers appeared frequently in the images. Future collaborations with researchers in other taxonomic fields are planned to better understand the disposition of ecosystems and biodiversity in the study area. Here you can find the references if you want to give a look. Well, I first want to thank to the organizing committee of the second Symposium of Women in Carcinology for this great job you're doing and the invitation to participate this year. Thanks to CONICET and UBA for the funding to do my PhD. And thank you for your attention. I look forward to hear your questions. Thank you.
Hi, Sophia. Can you hear me? Hi.
Yes, I can hear you. How are you?
Great. How are you? We love your work. It's very interesting. I'm biased. I love isopods. So we have a couple of questions for you in the chat.
Okay.
The first one is, do you intend to publish an illustrated catalog or checklist with our results, including an identification key?
Well, at first, it will be like a chapter of my PhD, complete one with the at least description of like morphological characters or characteristics of the new species we found. At least that. I think I will be equally like describing these new species in the future. I will try to do my best because I have a lot of things to do, not only describing and doing taxonomic studies. I will be also doing a complete phylogenetic analysis for a family. And I will be doing biogeographic studies too. So it will be like an work.
Yeah, that's a lot of work.
I will not be doing that, but I think I will do it in the future after I publish my PhD thesis.
Yeah, we had another question about is it easy to identify this species, or are there any identification keys that you use in the process?
Yeah, it does exist identification keys, but we also use like general references because sometimes these keys are old or we or need to be updated with new species that are coming really often to life.
Yeah.
So it's quite difficult, especially in some families. Some families have been studied more than others, and there are some families that are pretty diverse as Monopsidae, and it's really difficult to identify species in that family because we have like subfamilies, like 11 subfamilies, and then a lot of
that's very hard.
It's a hard work. So we may have more studies in some families that have been more studied as well. So it's like a bias. Yes.
Yeah, but it's great that your work is going to cover some of those
I hope so.
empty spaces. Oh, yeah, you answered this question a bit that someone asked if you used only morphology to identify the species or if you used molecular techniques as well, but you're going to, yes?
Yes, I'm planning to do it next year with a collaboration in Germany.
Yeah.
Probably, at least in this family, I will be doing this phylogenetic analysis because there's not only problems with general diagnosis. There's also problems with identifying a species as well, because you have problems with the diagnosis. Maybe we have cryptic species too, so we will need to do molecular at some point because of that reason.
Oh, yeah. So good luck. It's gonna,
Thank you.
there are gonna be a lot of amazing results, and we'll
I
be
hope so.
excited to see more from you in the future.
Okay.
Thank you very much, Sophia.
Thank you.
So now we're going to call our next presenter. It's Isabella Gonzalez. Hi, Isabella. Can you hear me?
Hola, ¿cómo estás?
Hola, hola, Isabella.
Hola. Ok,
Uh,
ustedes la pasan allá, ¿cierto?
okay, sí.
Bueno, entonces un saludo para todas, buenas tardes, me alegra mucho estar aquí con ustedes. Les voy a presentar un trabajo titulado El efecto de las macrófitas en la presencia y la estructura poblacional del cangrejo rojo Procambarus clarki en ecosistemas neotropicales. Este es un trabajo hecho en la Universidad Pedagógica y Tecnológica de Colombia y es en apoyo con los investigadores Jimmy Herrera y Yesid González Ruiz. Bueno, las macrófitas son uno de aquellos organismos que se encuentran en diferentes lugares acuáticos que pueden proveer lugares ya sea como refugio o como una fuente de alimento. Entonces, muchas de esas, de las estructuras que ellas tienen como tallos, raíces u hojas, dan una complejidad estructural que es conocida como complejidad fractal, que puede permitir que haya una intrincada rama que da refugio pues, a los organismos, ¿no? Entonces, varios organismos, dependiendo de su estado ontogénico, van allá ya sea para alimentarse, para desovar o para poder refugiarse en sus etapas de desarrollo. Unos de esos organismos que hacen uso de estas macrófitas son los cangrejos rojos, como pues, Procambarus clarki, ¿no? Para saber un poco de Procambarus clarki, esta es una especie que es nativa del sur de Estados Unidos y ella ha sido introducida en varios países del mundo y se ha convertido en exótica invasora en varias partes también, ¿no? Entonces, como ustedes pues, pueden ver más o menos en las imágenes, ella está prácticamente en Sudamérica, ha sido introducida en los años 80 y en Colombia se introdujo para 1985. Esta pues tiene varios estadios de desarrollo y uno de los más pequeños, pues como ustedes pueden ver la imagen, es muy pequeño, más que la palma de la mano, ¿no? Y pueden también en adultos ya tener entre 12 y 15 centímetros. Entonces, lo que nosotros queríamos ver era cuál era la relación que tenía Procambarus con las macrófitas, ya que ellos permanecen generalmente en la parte litoral. Para eso nosotros evaluamos la relación en el tiempo entre las macrófitas y los diferentes estados de desarrollo de Procambarus clarki en un ecosistema acuático altoandino. Pues el área de estudio es que esto es en la parte de Colombia, en los Andes, en la zona alta. Y pues lo que en el lugar donde nosotros hicimos el desarrollo de este proyecto es en un lugar que son unas lagunas de enfriamiento de una central termoeléctrica. En este lugar se produce energía a partir de la captación de agua, se pasa por unas turbinas y luego se libera agua caliente a estos lagos y pues las temperaturas normales allí son entre 10 y 13 grados, pero cuando hay influencia de esta empresa, el agua puede subir a 30 grados generando pues una cierta contaminación térmica que es aprovechada por organismos como Procambarus clarki, bueno, aprovechada porque él ha podido establecerse y subsistir y crear poblaciones en ese lugar. Lo que nosotros hicimos fue hacer una morfometría de longitud total, longitud del caparazón y longitud del cefalotórax, que es con la que se hace las cuestiones de las tallas, para poder hacer tallas. Pues para poder colectar los organismos se hizo un método de jameo en un transecto de 30 metros, donde se evaluaba que la macrófita evaluada estuviera un 70% aproximadamente de proporción en este lugar. Y ahí se colectaban todos los individuos, inmaduros, juveniles y adultos. Ahí las plantas seleccionadas, que fueron estas tres plantas, que fue Polygonum puntatum, Juncus effusus y Ludwigia peruviana, son las plantas que estaban generalmente en el litoral de estos lugares. Se valoraron también cuestiones como temperatura, conductividad de oxígeno, pH y sustrato. Pero ¿por qué se seleccionaron estas plantas que son del litoral? Es porque en la parte de la biología de Procambarus clarki es que él hace unas madrigueras en la tierra y es donde generalmente pasan su tiempo. Los machos luchando pues para poderse reproducir y las hembras en su lugar de protección y para desovar y para obtener alimento. Como resultado pues, de las tres plantas evaluadas, se encontró que la mayor proporción estuvo en Polygonum puntatum, en segunda en Ludwigia peruviana y en tercero en Juncus effusus. Entonces la mayoría de organismos estaban en Polygonum, que de hecho es la planta que tiene una complejidad estructural mayor respecto a las otras dos plantas que están allí, ya que ella tiene tanto, o sea, raíces pues hacia abajo sumergidas y además de eso una parte flotante muy amplia. Aquí en esta gráfica que muestro para ustedes están los 4 meses que se evaluaron y en las franjas grises es los tamaños de los adultos y en blanco los tamaños de los juveniles y los inmaduros. Bueno, como se puede ver aquí, por ejemplo, por mes, en mayo, casi todos eran adultos, como ustedes pueden ver, tanto en polígono, que es el rosado, el uji amarillo y el juncos, que era aquí el verde, casi todos pues en la parte de adultos. Luego llega julio, donde se empiezan a ver un pico de presencia de juveniles e inmaduros, y también en Ludwigia peruviana, y en juncos ya se encuentra solo pues adultos. Ya en la parte de septiembre se puede ver que hubo una temporada de reclutamiento, de desojo de reclutamiento, donde hubo una gran frecuencia de individuos juveniles y en Luguigia peruviana también, pero no igual en la misma proporción, y en Juncos siempre adultos. Y luego se puede ver en noviembre cómo disminuye la cantidad de organismos que son juveniles y pues de adultos también, pero pues se mantienen más o menos las proporciones en las otras dos plantas. Lo que podemos ver respecto más o menos a los resultados es que Procambarus usa las plantas de esta manera generalmente. Es polígono, que es la planta de mayor estructura, donde están como los individuos juveniles que lo usan principalmente como de refugio. Los adultos y juveniles en la parte de Ludwigia peruviana, que puede ofrecer también refugio, pero no igual que en polígono. Y en Juncos effusus solo se encuentran organismos adultos como de los organismos adultos, ¿no? Que ya que Juncos effusus no tiene una estructura tan, en la parte flotante no es tan compleja como en las otras dos plantas. Respecto a los sexos, prácticamente se vio como una proporción muy parecida, no hubo tantas diferencias. Aquí, por ejemplo, podemos ver en la discusión es por qué estarían los adultos en juncos. Es que se ha visto en otros trabajos que Procambarus permanece en plantas como juncos porque allá se encuentran organismos como los anfibios que hacen parte de su dieta, ¿no? Los renacuajos que son como una parte de las presas que él consume y que cuando hay Procambarus hace disminución de este tipo de poblaciones. También se ha visto en otras especies de Ludwigia, que es como Ludwigia grandiflora, que es una especie invasora en varias partes del mundo, en Europa, en Estados Unidos, que esta planta se ayuda con la presencia de Procambarus para hacer como su dispersión, o más bien tienen una especie de coadyuda entre los dos, ya que Procambarus pues, se encuentra entre Ludwigia y hace uso de los recursos, y pues Ludwigia pues se dispersa. Aquí, por ejemplo, se pudo ver cómo cuando hay macrófita, aumenta el número de organismos de Procambarus clarki, y además de eso no solo aumenta el número de organismos, sino también las fases de Procambarus que se encuentran ahí. Como ustedes pueden ver en la parte de la derecha, entonces aumenta tanto los individuos juveniles inmaduros como pues, los adultos, como nosotros también pudimos ver en esta especie que era peruviana, pero que tiene más o menos unas características parecidas. Entonces se ha visto que esto es un comportamiento de Procambarus, como usa este tipo de planta. Y también hemos podido ver respecto a los tamaños que se tomaron en cuenta para las morfometrías de ese lugar, es que aquí en la parte altoandina, Procambarus tiene 2 picos de desuva. Yo les muestro a ustedes datos de el segundo semestre del año, pero hemos visto que también se ha visto para el primer semestre del año, donde tiene 2 picos de desuva que están relacionados con los regímenes climáticos en la zona altoandina, que son un régimen bimodal, ¿no? 2 picos de lluvias al año. Y se ha visto que en Europa, por ejemplo, puede haber hasta 3 picos dependiendo de los lugares y en Estados Unidos también diferentes. Entonces, lo que hemos visto es que está regulado por el régimen hidroclimático, ¿no? En este caso, como se ha visto también en otros documentos. También lo que quería mostrar para ustedes es que en muchos artículos, en muchos pues, documentos habían tenido algunas dificultades para poder hacer evaluaciones de Procambarus juvenil e inmaduro, y pues con el método que nosotros usamos, pues lo creemos recomendable cuando uno quiere hacer evaluaciones ecológicas de este organismo, porque podemos colectar organismos muy pequeños y hacer pues, las evaluaciones para verlo de desova. Entonces es un método recomendado el que se usó. Y muchas gracias a ustedes, muchas gracias por escucharme y por estar aquí.
¿Tú puedes escuchar ahora?
Escucho.
Ay, bueno.
Disculpen, yo sé que tuve unos inconvenientes en la parte de
No hay
inglés,
problema, no hay
entonces.
problema. No hay problema, hijo. Nosotros tenemos una pregunta para usted aquí.
Ujú.
¿Cómo haces los mostreos? O mostraste unas fotos, pero ¿alguna vez usaste trampas para mostrar los procámaros?
Sí, hay unas trampas que existen para las redes cangrejeras que es muy común que parecen estas que son Z real o Accessotech, que parecen una sombrilla que tiene diferentes entradas, pero lo que nosotros vimos es que ella resulta que solo capturan adultos y además de eso, generalmente los machos, que son los que tienen más el instinto de atacar e ir por el alimento, en este caso para esta especie. Entonces era un método muy subjetivo para un tipo de talla. Y cuando nosotros queríamos evaluar dinámica y una cuestión poblacional, siempre teníamos falta de los inmaduros y los juveniles más pequeños en naturaleza. Entonces, cuando nosotros usamos este método que es un transecto y pasamos caminando durante 10 minutos, jameando, que es nuestra palabra en español, para ellos macrofit sweep, que es la forma en que podemos como retirar todos los organismos de todos los tamaños, porque el poro de la malla es de 5 milímetros. Entonces
Oh,
ahí no se nos
ok.
queda nada.
No queda nada así, ¿verdad?
Ujú.
Nosotros tenemos una otra pregunta para usted. Sí, ¿cómo es una especie invasora? Si él está a competir con alguna especie que está a vivir natural en la región y puede traer algún tipo
Eh,
de males.
pues nosotros hemos visto pues en varios documentos de que él puede ayudar a transmitir un hongo para para otros cangrejos nativos. En este momento se me olvida el nombre. Ah,
Pero,
una fanomisis es el hongo. Entonces, digamos que ese sería el que se transmite a unas especies nativas que sí se ha registrado. Y además de eso, un Procambarus es muy agresivo. Entonces, lo que hemos visto es
sí,
que
sí,
él
¿verdad?
como que este comportamiento genera como un desplazamiento de las especies nativas. Sería bueno comprobarlo en laboratorio, ¿no? Con esto, pero lo hemos visto ecológicamente y cuando hicimos nuestros muestreos vimos que había ese tipo de competencia. Y pues no, es un omnívoro que consume cualquier cosa. Entonces también
Claro, es
sucede esto.
verdad. Yo también tengo una duda, porque yo creo que en algún momento yo tenía visto que algunos procámbaros tienden a tener alguna especie de partenogenia. ¿Esto es verdad? ¿Tenía algo que puede hacer con procámbaros claro que también? Porque tú mostraste muchos machos en hembras, pero puede tener en algún momento algún comportamiento de partenogenia Pero las hembras, yo creo que sí. Yo creo que
Pues
no, no sé.
nosotros hemos visto artículos que sucede como con unos querax, con otros de la familia de los cambaridos, si no estoy mal. Y pues que puede suceder esto, pero en lo que nosotros hemos visto es que la reproducción sigue siendo como más de la parte sexual, ¿no? Y pues
Ok.
las hembras tienen su cuidado parental y todo esto. Que puede llegar a suceder porque pues lo hemos visto en varios documentos que lo relacionan, que puede suceder, pero hasta el momento no lo hemos visto. No, pues nosotros no, pero parece que sí puede suceder. Pero en este momento Procambarus ha mostrado comportamientos totalmente sexuales, o sea, de reproducción
Porque yo
sexual.
veo, para esta especie todo es posible.
Sí, las especies invasoras van a hallar la manera de hacer lo que sea por sobrevivir, están adaptadas a eso.
¿Verdad? Isabela, muchas gracias por su presentación. Yo creo que no tenemos más preguntas,
Muchas gracias a
pero
ustedes.
mucha suerte en los próximos trabajos. Gracias por presentación.
Muchas gracias.
Chao. We're going to call the next presenter. The next presenter is Kaori Wakabayashi. Hello, Kaori. Can
Hello.
you hear me?
Yeah, I can hear you. Can you hear me?
Yes. We're going to
Okay,
put
thank
your
you very
slides
much.
on the screen.
Yeah.
Feel free to present whenever you're ready.
So this is the one that I just put it. Or
Yeah.
is this?
Yeah, it's yours.
Yeah.
I'm
Okay.
pretty sure you're gonna be able to.
Thank you. Yeah. Thank you very much for the introduction. I'm Kaori Wakabayashi from Hiroshima University, Japan. Today I'm going to talk about the unique planktonic shrimp, Amphionides reynaudi. Actually, this work was led by one of my students who graduated from a university in last March. Okay. So a unique planktonic shrimp was found in 1832. And then this famous taxonomist Milne Edwards give the name Amphion reynaudi to this species. And a similar species were found later. Two of them are described as a new species, but nowadays we consider these are the synonym of Amphion reynaudi. In 1904, Zima found another form of this and named Amphionides perdiviae. And then 30 years later, Gurney had a chance to examine this second form, and then he considered this second form actually the same species and different development stage of the first one, which is Amphion reynaudi. He considered that this is post-larva of this form. And then later, Heegard also had a chance to examine actually the Dana expedition specimens, and then he agreed Gurney's idea, and then he also think that this second form should be the post-larva or adult form of the first one. And Williamson, who examined the second form, considered that this is the adult form of the first one, Amphion reynaudi. And actually, the genus name Amphion is used in the insect. That's why he applied the Amphionides as the genus name for this species. That's why we used Amphionides reynaudi at this moment. Because of this unique morphology, Williamson considered that this species should be placed in outside of the decapod and then give a new order, Amphionidesia. However, the recent molecular techniques show that this species should be placed in Calidia, particularly in the Pandaloidea. So, Williamson proposed that this is an adult form of Amphionides reynaudi because he found that ovary of this species. But researchers doesn't really support his idea because of the two reasons. One is he reconstruct a single form using 43 damaged specimens. So researchers doesn't really think that these ovaries are actually from the Amphionides. It can be from other species. And the second reason is no one actually ever seen this Williamson's observation after his publication. So so that's why adult form of the Amphionides are considered unknown. So we are interested in finding what is the adult form of Amphionides reynoldi. That is our final goal. But question is, did anyone confirm the genetic identity between the two forms? Because these two are originally described as a different species. But so far, no one examined about that. And to do this, we need a DNA workable specimen, but where we have it, actually we don't have any DNA workable specimens because the latest record were made 50 years ago. So we were so lucky to obtain freshly collected adult form of Amphionides reynoldi from the Negashima Islands, Japan. We did mid-water net towing of the Negashima Island and then found this specimen. So we tried to observe the morphological features of these specimens to compare with previous description, and also tried to extract the try to extract DNA from the freshly caught specimens to compare with the larval genetic information. Actually, the larval form of Amphionides reynoldi is not so difficult to correct around Japan. So we collect the larval form also as well to compare the genetic information of the adult form. So adult form is very rare. So far, documented records are all here. So mostly found from the west coast of Africa, but some records are made in the Indian Ocean and even the Southeast Asia. And our collection was the first time from around the Japanese waters. So this is the morphological comparison of among the records, including the original description and ours. So the specimen size are mostly the same. And the carapace, some say that cylindrical, and then some said oval. And our specimen has the carapace shape of oval, but because this is caused by the damage from the collection time, the carapace was somehow open. That's why it looks oval. That's why we think that the comparison of the carapace doesn't really work for morphological comparison. This adult form show a special, very unique character in the anterior part of the body and the preopod one. So as Hegard described and illustrated, the anterior part of the body of an adult form of Amphionides is like this. It has short peduncle of the antennulae and then wide and flattened exopod of antenna. And the rostrum is short and shaped triangle. And then eye also, it's very unique features. Hegard pointed out that there is a photophore here, but we cannot confirm the presence of photophore, but other features are mostly the same. And preopod 1 also very unique. Preopod 1 of this specimen is flattened, very long, and surrounded by the setae. These characters are agreed with the previous description. So we conclude that all records are morphological identical. Only the problem remains is that Williamson pointed out the presence of ovary, but none of researchers, including our specimen, cannot confirm the ovary presence. So our molecular phylogenetic analysis show that adult form of the Amphionides reynoldi form a clade together with the larval form collected from the Japanese waters and the water of Canary Islands, which is in Spain. So the genetic distance among these four specimens are almost the same as the genetic distance within the species in Heterocarpus and Cipher and the Physetocallis species. So we conclude that these four specimens are the same species. So genetically identical, sorry. That's why we conclude that these four species can say that it's the same species. And some researchers pointed out that the Amphionides and Physetocallis look similar, so it can be close related. However, our molecular phylogenetic analysis didn't really show the close relationship with these two species. And our molecular phylogenetic analysis also showed that the Amphionides reynaudi is nested in the Pandaloidea. So Pandaloidea has two families, and one of the families are now classified into 5 different groups, 5 different genetic groups. And we wanted to show that Amphionides belong to which groups or within the Pandaloidea. However, this time our molecular analysis failed to show it. So that's our next goal. We are trying to improve our molecular analysis by adding more genetic information, including like a protein coding genes. So overall, our work concludes that larval form and adult form are actually genetically identical. So both are now, we can use the name Amphionides reynaudi for both forms. But this does not mean that adult form is a true adult, because we haven't confirmed that the maturity of this one. So our next step is whether we can collect life of adult form to confirm the presence of ovary, or we may try to culture the larval form until the adult form. We actually tried to culture the larval form and then obtained successfully several larvae metamorphosed into this form. But this form was different from adult form. So we consider that this is a post-larval stage. But we confirmed that it works, this method works. So next time we try to collect the larvae from the wild and culture in the lab until adult form to achieve a conclusion whether the adult form develops an ovary or not. Yeah, that's all. Thank you very much.
Thank you, Dr. Kaori. This is very interesting. It's very, very weird looking, this shrimp. It has like this very big carapace. I thought it was very interesting. So many questions in the chat. How many adult specimens did you collect around Japan?
Yeah, so far we could only one specimen, a single specimen. But
Yeah.
I show you the record. We may be able to collect the adult form every 4 decades, like that span. So far, I tried to gather all the records starting from the 1904.
Yeah.
Only 4 or 5 researchers describe
Oh my God.
the records. So I may need another 20 years to have another specimen.
Hopefully not.
Hopefully we can get earlier.
Yeah. Another question is, what part of the larva did you use to extract DNA? What about the adult?
Yeah, so we use the abdomen muscle for both.
Oh, for both. Okay.
Yes.
Yeah, it seems difficult to find them. Yeah.
Yeah, we will try.
Yeah, and do you think the larvae development is extended to explain this large distribution?
Yeah, someone collected the larvae from the wild, and then the larval distribution is quite wide. So this time I used the larvae from Japan and then larvae from the Canary Islands. But I'm sure that the larvae are also distributed in your place, like off Brazil or Caribbean Sea. I'm very interested in including those specimens to see whether genetic distance is far or they are cosmopolitan or not. So I'm welcome any collaboration from Brazil
for
or
sure.
Caribbean. Yeah.
Yeah, this is very interesting.
Yeah.
Do you believe there is spatial segregation between adults and larvae? Why so hard to sample adults?
Yeah, that's possible. It's the people say that adults distribute in the deep water, but the larvae can be found from the shallow, although larvae also can distribute the deep water. We are not very sure. Our record was from 900 meters, but that is the almost shallowest from among the old records. So like what people say, the adults can be distributing only in the deep water. We are not sure, but probably like that.
Yeah, explains why it's so hard to find them.
Yeah.
Thank you very much, Dr. Kelly.
Thank you
It
so
was very
much. Thank you
interesting.
so
Thank you
much.
for your talk. So now we're going to call our last presenter for the day, Ananya Chakraborty. We're going to play her video presentation, and then we're going to call her on the stage to answer questions.
Everyone, it's an honor to be here today at Second Symposium of Women in Crustacean Science to share my work with such an inspiring international community. I would like to thank the organizers for giving me the opportunity to present my research here. I am Ananya Chakraborty from Bangladesh. Today I will be presenting my research entitled Microplastics and Cadmium Chloride Co-Exposure in Female Tropical Freshwater Crab Sartoriana spinigera, evidence from growth, molting, hematobiochemical indices, behavior, and histomorphology. Microplastics, which itself is a toxic compound and closely interact with other contaminants, particularly heavy metals. Due to their highly hydrophobic nature, microplastics readily absorb heavy metals, forming harmful complexes. When they enter into the body of organisms, they cause potential physiological damage. This experiment was conducted with two different plastic polymers. One is polystyrene, which is known as hazardous polymer, while another one is polyethylene terephthalate, as it is widely used plastic. Besides, cadmium was selected because of its mutagenic and carcinogenic properties. There might be a question why this species has been selected. Because in Bangladesh, there is no prior toxicological study on freshwater crab, particularly on this species. Additionally, it is crucial to aquatic food chain, has nutritional and ecological importance, survives in harsh environmental conditions, and lives close to water and sediment, which make it a good indicator of contamination. The aim was to compare the individual and combine
Hi everyone, I'm sorry, we're having some connection issues, I think, so the video is going a little weird, so we're going to restart the video and hopefully it will go smoothly now. I'm so sorry for the issues.
microplastics. Good afternoon, everyone. It's an honor to be here today at Second Symposium of Women in Crustacean Science to share my work with such an inspiring international community. I would like to thank the organizers for giving me the opportunity to present my research here. I am Ananya Chakraborty from Bangladesh. Today I will be presenting my research entitled Microplastics and Cadmium Chloride Co-Exposure in Female Tropical Freshwater Crab Sartoriana spinigera, evidence from growth, molting, hematobiochemical indices, behavior, and histomorphology. Microplastics, which itself is a toxic compound and closely interact with other contaminants, particularly heavy metals. Due to their highly hydrophobic nature, microplastics readily absorb heavy metals, forming harmful complexes. When they enter into the body of organisms, they cause potential physiological damage. This experiment was conducted with two different plastic polymers. One is polystyrene, which is known as hazardous polymer, while another one is polyethylene terephthalate, as it is widely used plastic. Besides, cadmium was selected because of its mutagenic and carcinogenic properties. There might be a question why this species has been selected. Because in Bangladesh, there is no prior toxicological study on freshwater crab, particularly on this species. Additionally, it is crucial to aquatic food chain, has nutritional and ecological importance, survives in harsh environmental conditions, and lives close to water and sediment, which make it a good indicator of contamination. The aim was to compare the individual and combined effect of microplastics and cadmium on growth, behavior, and bioaccumulation, also to observe the hematobiochemical and histological changes associated with microplastics and cadmium toxicity. The experiment included 5 treatment groups and 1 control group. It lasted 14 days and involved bath treatments using the female live crabs in each tank. I went through this methodology. For mentioning the outcomes, I would like to start with behavioral changes. After 7 days, there was no significant alterations. After 14 days, quite few alterations were noticed, such as locomotion was reduced as they got stressed due to the toxics. Higher survival rate and SGR were found in control group compared to the exposure group. Polystyrene caused higher microplastic accumulation in gill, whereas polyethylene terephthalate caused higher accumulation in hepatopancreas. Noticeably, microplastic influence comparatively higher cadmium accumulation in combined doses rather than single dose. Hematobiochemical parameters. ALT and AST indicate liver condition. Compared to the control group, ALT was elevated throughout the exposure group with highest influenced by cadmium, and AST was elevated by polystyrene. Total cholesterol found to be increased in all exposed groups in contrast with control, but PET and its co-exposure group documented higher impact. In case of triglyceride, the impact of PET is clear. Total protein was highly affected under cadmium and polystyrene. Higher glucose level indicates stress condition. Elevated glucose levels in treatment groups compared with control confirms stress condition due to the presence of toxicants. Brix, which is a traditional tool widely used in lobster and shrimp industry, it estimates the percentage sugar content of aqueous solution. Several studies outlined the Brix value peaks just before molting. Here, maximum value was recorded in PET. Brix used to confirm the molting tendency. In this experiment, PET induced successful molting, whereas due to molting inhibitory nature of cadmium resulted in incomplete molting in case of PET plus cadmium exposure. Here comes an interesting part of this experiment, a video of successful molting. Luckily, I could capture during trial period molting induced by PET. Principal component analysis makes the key findings easier to understand. Here it is clear that the result is not particularly conclusive for any toxicant. The effects of toxicants were dose dependent and varied through different parameters. Survival rate and SGR positively correlates with control, which is usual as there is no effect of toxicant. PET and its co-exposure impacted Brix, total cholesterol, triglyceride, and glucose highly, whereas cadmium and its co-exposure highly affected total protein, AST, and ALT. There are some histopathological abnormalities of gill marked in exposure groups by comparing them with healthy condition of control. Here, except control, all treatment groups reported deformities of hepatopancreas and mostly affected by PET-associated group. And very lastly, the key findings. However, the growth and survival rate were dose-dependent, highest mortality in polystyrene, significant alteration in biochemical parameters, PET-induced molting, and cadmium inhibited molting with co-exposure with PET, comparatively higher sensitivity of polystyrene and co-exposure was recorded in case of histomorphology. I want to acknowledge these institutions and laboratories for their support, and also my manuscript of this work is under review in the journal Ecotoxicology and Environmental Safety. Thank you all. If you have any query, feel free to ask me. I will try to answer the questions.
Hello, Ananya. I'm so sorry
Hi.
for our connection issues. Sorry for having to play your video again. But your work is very interesting.
Thank you.
The results are very nice. We have a couple of questions for you. How long does it take to finalize the mold? It's written the
In
mold,
case of
but it's the
molting,
mold.
it takes 10 days.
10 days?
10 days
Oh.
I found the first molting, and the video I have provided is from the day 12.
Oh, okay. Yeah. Do you need a big portion of the gills to make the histological preparations?
No, I didn't take much portion of gills. I took only 2 or 3 filament for the histological preparation, and
Oh,
I
yeah, that's good.
each crab from that treatment,
Mhm.
and that's why I didn't took much portion.
Okay, yeah. Any more questions?
I have a question.
Yeah.
Sorry. just because I'm next to Amanda. And do you need to do some special preparation to do the slides with the gills? And which coloration process do you do?
In case of histological preparation of gills, I faced the difficulty that compared to other muscles, the gill muscles are too hard, and I had to go for a longer filtration period, infiltration period for the making the blocks of histological.
And the infiltration, you do the room temperature?
Temperature, I have done the infiltration in the hot air oven, and the temperature maintained around 80 degrees Celsius.
Oh, okay. So interesting. Thank you very much, Anai.
Thank
Thank you,
you.
Ananya. Very nice
Welcome.
work.
Thank
Any
you.
more questions? I don't think there are any, but thank you very much for presenting your work.
Welcome.
Bye, Ananya. Thank you. Thank you, everyone who presented your work today and everyone who watched us and asked questions. We're sorry for the few technical issues we had, but thank you, everyone who stayed with us and watched the nice presentations. All the women who were here today with us presented. And we're still having a few presentations over in the Spanish session if you want to switch over to there. If not, we are going to come back tomorrow. Tomorrow morning, we have more presentations. Tomorrow will be in Portuguese, but we have more presentations tomorrow if you're interested. And then at the end of the day, we have the closing ceremony where we'll have the share about the prizes and who won. So please come tomorrow as well. And we're going to be waiting for you tomorrow. Thank you, everyone.